So I'll go relatively quickly through this first part, hopefully, because there's the full video online that has all of these slides in it, up to the helmet slides. The first pathogen we talk about is malaria, which is the single most important pathogen from a parasite perspective in terms of human health and disease, with 1 to 2 million deaths per year. And the distribution of the parasite really determined by the distribution of the malarial vector, which is a species of mosquito, several species of mosquitoes. The malaria life cycle is quite complex, as noted, has been modeled many, many times using relatively simple differential equations and then many more sophisticated approaches. But there's a mosquito phase and a human phase. And within parasites, this is a lot of how people frame their thinking around parasitic infections, which is intermediate hosts versus definitive hosts. The definitive host is always where sexual reproduction occurs if it occurs for a particular parasite. So in the mosquito actually is where the gametocytes are picked up from the blood. So the sexual forms of the parasite from the human, where they'll then mate in the mosquito, form an egg and oocyst that will burst and release a lot of the infectious form of the parasite, which is called the sporozoite, into the jaws of the mosquito, which will then be available to the blood when the mosquito takes another blood meal. The sporozoite then goes immediately to the liver cell very, very fast. This process takes probably a matter of minutes at most, potentially even less than a minute. And the importance for thinking about that time frame is that this is the window in which the parasite would be available at its lowest concentration to the immune response for targeting, particularly by antibodies. It goes into the liver cell. It then starts replicating in the liver cell to very, very high levels. It then kills the liver cell by bursting out of it, releasing the blood stage of the parasite, which is really the disease-causing stage of the parasite, which will now infect the anucleated red blood cells. So your eukaryotic red blood cells, your mammalian red blood cells, I should say, do not have nuclei, will replicate within the red blood cells, again, to very, very high levels, destroying the red blood cells, which is one of the major causes of the disease, is the anemia caused by the depletion of blood, as well as the inflammation around the replication in blood cells. Stochastically, a subset of these ring stage parasites will differentiate into the gametocytes. Not all of them. Again, it's a stochastic process, and they'll be available in the blood for the mosquito to start to cycle again. There are a lot of innate patterns in the malarial parasite, which is called Plasmodium, that can be recognized by the innate immune system. And so the lipid anchors that are used to attach many of the surface proteins on the parasite are recognized by the TLR system, and the TLRs are probably quite important in malaria. There are also metabolites of malaria, like isopentyl pyrophosphate, which are a target for one of the innate T cell subsets that we talked about, the gamma delta T cell. We talked about it very briefly, but I think it's one of the main purposes of gamma delta T cells in the blood of humans is actually to recognize malaria. And this gets to the sort of evolutionary importance of malaria for human development. So once you have this initial activation of the innate response by TLRs and other innate receptors, it's going to drive all of the types of type 1 responses for intracellular parasites that we talked about yesterday. The thing that gets a type 1 response going, one of the cytokines is called IL-12. And that'll lead to the differentiation of your T cell to a type 1 Th1 cell, which is characterized again by interferon gamma, IL-12. These are the classic cytokines of that response. They're going to drive the production of CD8 T cells and other effectors to try to go out and kill the infected parasites. So there's a lot of work done on malaria in mouse models, like some of the pathogens we've been talking about. No mouse model is perfect. I have a lot more information on this in the online talk. But we have learned some about how mammalian immune systems interact with malaria from the mouse models. And so you can have a model where you have normal control of the parasite, which is what happens in most humans that are infected. If you knock out gamma delta T cells, there's maybe a slight delay in clearance, but it's not absolutely essential. If you knock out CD4 T cells, which again are these critical organizers of the entire adaptive immune response, then you fail to clear the infection, and you get this cyclic parasitemia, which corresponds to this cycle. So this is somewhat synchronized. You see this in human infection as well, where you get these waves of fevers because all the parasites are going into your blood cells, the inflammation drops down a little bit, then they all burst out within a very short window. You get a massive inflammation, you feel terrible, and then they all infect another round of red blood cells, and you feel maybe a little bit better for a little while. If you get rid of B cells, there's a bit of a loss of control, but it's not as dramatic as CD4 T cells. If you get rid of interferon gamma, which is that critical cytokine that mediates the whole type 1 response that's going to get rid of intracellular pathogens, then the animals don't survive the initial infection. And there are various forms of malarial pathology. And again, there's more of this online, so you don't just die from the consumption of your red blood cells and from the inflammation. There are special forms of malaria where mostly all associated with immunity, though. So the reaction of the immune response causing inflammation and causing mechanical blockades in your vasculature can cause cerebral malaria and placental malaria. And then, of course, severe anemia is a critical component of the disease. From a modeling perspective, malaria is an interesting one to think about from a within host, between host model, because vector control, the control of the mosquito, is really critical, and it's the way that in countries that have successfully controlled malaria that it was successfully controlled. So for instance, in the US, we used to have malaria all the way up through to Boston. So if you've been to Boston, the fens around Fenway Park used to be riddled with malaria. Here where I live in Memphis, we have a big bridge that looks like an M that people say could stand for malaria because malaria used to be a huge problem here. And the only way it was really controlled was through getting rid of the large reservoirs of mosquitoes. And so modeling of infectious spread with the mosquito as the transmitting host has actually helped a lot, I think, in terms of the public health efforts and determining what are the most effective methods, particularly bed nets, because these are literally nets you put over your bed that the mosquitoes can't get through, and particularly bed nets that are soaked in pesticide. Because a lot of the mosquitoes that transmit malaria bite at night. And so if you can lure them to a situation where they will kill themselves right at the moment where they'd be most infectious is much more effective than just trying to get rid of mosquitoes. It's much more cost effective than trying to get rid of all mosquitoes directly. There are also some very crazy approaches that are less and less crazy each year, because they're actually being tried, where they release genetically resistant mosquitoes to malaria, because the malaria costs the mosquito a bit too. And so mosquitoes that would be intrinsically resistant to malaria would probably have a replication advantage. There's a lot of work on vaccination for malaria. And again, here's where modeling, I think, is really potentially very useful, because there are 3 very distinct stages of the malarial life cycle that could be targeted by a vaccine. A lot of the vaccines, because they comport, I think, with our notion of what a good vaccine would be, target the liver stages. And so the idea of trying to block the block the parasite from getting into the liver cell or from getting out of the liver cell. That's when the colony is at its smallest. And so if you can eliminate it there, you're in a good situation for blocking the rest of the infection. But this is the hardest stage to do that, because you have such a short little window in which the parasite's exposed in the bloodstream where an antibody might work. And then the liver is kind of a tough environment to mount an immune response in, because it's a generally tolerogenic environment. So getting CD8 T cells to go in and kill in the liver can be tough. There are ideas to vaccinate against the bloodstream stages, which have all very different proteins on them from the liver stages. And so they would have to really be distinct vaccines, but this would not prevent infection. It would only prevent disease, which would obviously be a very important goal. But I think it's gotten less focused because there's you just like the idea of blocking the infection entirely. And then there's something called the replication targeted vaccine, which would actually just target the gametocytes, which are very antigenically distinct. The parasite seems to have invested a little bit less in its ability to evade the immune response against the gametocytes, so the sexual stages. And this would do nothing to protect the person that has actually been infected with it, but it would protect them from infecting anybody else. So this is often called an altruistic vaccine, and the thought that it would be maybe combined with one of these other vaccines, because many people maybe won't sign up for this on their own. There is an approved vaccine now for malaria, that's RTSS, and the initial report from it was incredibly positive. So this was in an infant cohort in a malarial prone area. This is an NEJM paper from almost 8 years ago now. And you see, like our SARS-CoV-2 vaccines, this looks not quite as good as those, but pretty good, and the best thing anyone had seen for malaria. And this is a protein that's on the sporozoite, so the stage that infects the liver. So classic vaccine, you're just trying to get rid of that stage. And it's combined with an adjuvant. So the immunologist's dirty little secret to activate the innate immune response. This is a lipid adjuvant, AS01. And every time they redid a trial after that, though, it kind of started looking less and less good. It is still in use and in various other trials, but it went from 61% effective to 30% effective. Given the enormous burden of disease of this infection, though, it's stayed moving forward with various research. It also has different efficacies in different areas because there is some variation among likely the health of the population that's being infected, and then also the parasites that they're being infected with. So I just end with that, just mentioning that transmission models have contributed substantially to our understanding of malaria control. Within host modeling, it's crucial to determine the potential efficacy of these 3 types of vaccine candidates. We didn't talk much about this, but I talk about it in the online video, but there's real threshold effects of malaria infection. And that, and you'll see this for the helminths as well, if you get repeatedly infected, you actually start building up immunity. And if you do get infected again, it's a very mild infection. And so there's a very common epidemiological observation that people that grow up in endemic areas develop a pretty good resistance to malaria. And then if they go to a non-endemic area and live for a very, very long time and then come back and get infected again, it can be a very severe infection because they've lost that protective immunity that they had built up over time.